We good? Dom, you on? Ready?
Yeah, I'm ready.
Cool. You ready, Tori?
1 second.
Okay.
Okay, I'm ready now. I just had to see what time it was.
Alrighty.
Okay.
And you can see our screen and stuff?
Yep, I can see everything.
Cool. So the title of the paper that Dom and I chose is Protonation of Piezo1 Impairs Cell Matrix Interactions of Pancreatic Stellate Cells. And the reason that I chose this was because I really enjoyed the first paper that we did in this class. It kind of introduced me to like how cells act as more than just chemical processes, but they're mechanical processes as well. So I liked the analogy that you made, Tori, to the milk carton. And so when I was scrolling through looking for a paper and came across the Piezo1 aspect of this one, I thought it was going to be a really cool opportunity to like go forward in the understanding of piezo and like what more has been found out about it. I think a really cool aspect of science is like you learn stuff and then there's always more stuff to learn. So since we had like some base of Piezo1 knowledge, I was really excited to go further and learn what other scientists had found out about it like more recently.
So relating this paper sort of towards disease or whatever, so they were like focusing on pancreatic ductal adenocarcinoma or PDAC. So PDAC is like very bad cancer. I don't want to say bad cancer because all cancers are bad, but so like 90% of pancreatic cancers are PDAC, so it's like very common in terms of in relative to other pancreatic cancer, and it develops from the cells lining pancreatic ducts. So you can see on the right, there's like a little graphic, and there's like the acinar cells in the bottom right, and those duct cells. So they often sort of like accumulate and like agglomerate or whatever. And so PDAC is very difficult to treat or cure. So like the only way to cure PDAC is to remove the tumor. But in 80 to 90% of the patients that like have PDAC, like the moment that they notice that the patient has PDAC, it's too late to like surgically remove it or cure it. So for that reason, like there's a very high death rate for people with PDAC and very low recovery rate. So in 2014, kind of a little outdated, but still close enough. So like there are 46,000 new cases, but 39,000 deaths. So a lot of deaths in relation to the number of cases. So this tumor or like these tumors in PDAC are characterized by like a high pressure in a very acidic environment. So they kind of wanted to study how these two factors can like affect these cells, these like PSCs, pancreatic stellate cells. And they also know that in these cells, like calcium signaling plays like an important role for their physiology and pathophysiology. And because piezo is like an ion mechanosensitive channel, they wanted to study like the relation of piezo1 in these cells and whether it affects their development or any other aspects of the cell.
Yeah, so now we're going to jump into the figures. They started out by establishing their models of Piezo1 in the cells. So in the PSC, the pancreatic stellate cells, with this figure, they were just showing that Piezo1 is functionally expressed. And to do this, they used a manganese quench experiment, which is essentially in the green here. This is a fluorescent marker of calcium. And then they use the manganese as a kind of a simulator of calcium influx. And so when Piezo1 is active, manganese enters the cell here and quenches the activity of the fluorescence. And then that readout that you get from the loss of fluorescence, it translates into a rate of influx. And so you can, since it's so similar, the manganese and calcium let in by Piezo1, you can use that to infer piezo activity. So what we saw here is that with the Piezo1 activator Yoda1, there's a much greater influx of manganese and therefore calcium. and you see that quantified here on the right. And then they also set up a model in transfected hex cells, where they transfected hex cells with Piezo1 and activated them with the Yoda. They did the same. They transfected the hex cells and didn't activate them. And then they used hex cells transfected with GFP and then treated with Yoda, and then hex cells with GFP that weren't treated. And you see that they did get some amount of manganese and calcium influx here. And these are just shown because they used them in later experiments, and later on, since they've set this up, you can just assume that, they assumed that the Yoda 1 addition translates to activation of Piezo1.
I have a question about Yoda 1. Is Yoda 1
Yeah.
with this like cytoplasmic, or is it membrane? Like what kind of protein is it? I might have missed it.
I'm not 100% sure. I know that it is, I would imagine that it's an extracellular
Like an agonist.
activator.
I don't know if it's a protein, but
Is it, yeah, it's an
some sort of
activator
agonist.
of Piezo1. I don't know if it's cytoplasmic or extracellular though.
Yeah, or it's not membrane though.
It
doesn't matter. Cool.
I don't believe so.
Cool.
Yeah.
If you don't know, it's all good. Cool.
But then they do the same thing where on the left you have the readouts, and then on the right they quantified it a little bit. There we go.
Okay, so now like they established that Yoda 1 is, or not Yoda 1, Piezo1's like expressed on these PDAC cells, and then Yoda 1 can activate them to lead to like the influx of calcium. So they wanted to see like what might be the like consequences of this activation and then influx. So like canonically, like calcium influx can lead to like phosphorylation changes in cytoskeleton, like myosin 2 and stuff. So they thought maybe like motility might be changed. So when they tested motility, so you can see in A, they have control, and then they also have Yoda. So like in control cells, there's like some movement of these cells. So this is like a 2D saying of like motility. And you can see, yeah, in A. So the control, there's like some movement, versus Yoda 1, there's like a lot of translocation. And then in B, they're showing a quantification of this. So on the left, they're showing velocity. So they're showing in velocity, there's a significant difference in speed, but not super drastic between Yoda and the control cells. But with the translocation on the right, you can see there's like a very large difference in how far they travel from the center. And furthermore, they show different doses of Yoda 1 in both of these. And you can see that, especially on the right with the translocation, that control compared to 5 micromolar Yoda, there's a very large difference in translocation, versus 15. There's still significant. When you pump it all the way up to 50, there's no difference with control. So then it's suggesting that this is like a dose-dependent response. And so getting back to like the acidosis, so we mentioned earlier that PDAC, like these tumors or like large conglomeration of cells, is like characterized by high acidity, or low acidity, sorry. So like acidosis. So they're testing how this acidity possibly affects Piezo1 function. So on the left here, they just have control with GFP transfected cells. But on the right here, we're showing different intracellular and extracellular pHs. So you can see at the bottom, you have like those red and blue circles and rectangles. So like that circle is a cell, and inside of that would be intracellular pH, and outside would be extracellular. You can see that the blue area would be more basic conditions, not basic conditions, but like normal physiological pH, versus the red would be pH of like 6.6, so like an acidosis, representing acidosis. And you can see that in the entry rate of manganese or calcium, which is what it's supposed to show, is that in these regular control cells, you can still see there's a large increased entry rate, versus when you change the extracellular pH to lower, you can see that this entry rate is slightly inhibited, but when you change the intracellular, it's inhibited even more, so it has more of an effect. So they're showing this intracellular pH is more important for inhibiting Piezo1 function. And then when you combine them both, there's even slightly more. It's hard to tell, but it's slightly more inhibition. But they also test much less cells compared, because like there's 129 versus 34, but you can still see there's inhibition of Piezo1 function.
So can I just review out loud for me? So the acidity internally or externally is reducing the flow of ions through piezo.
Yes.
Yeah.
And then the flow of ions through piezo positively affects motility. Is that correct?
Yes.
Yes.
Okay. Okay, cool. So we would like be making our cells more kind of stable and place here. Okay, cool. Thank you. It's interesting.
And then they also tossed in a couple of experiments where they looked at the effects of the cytoskeleton on Piezo1. So they brought back our old friend blebistatin, and you can see that when treated, the cells have less of an influx of manganese and calcium. So that's showing that in A here. So that's showing that perhaps the Piezo1 activity is somewhat dependent on myosin activity, since the cells treated with Yoda1 and blebistatin have a significantly lower entry rate. And then in this other figure, they're showing that the intensity, so they're showing that the intensity, the intensity of myosin increases in the Yoda1 treated cells as opposed to the control cells. So when the piezo is active, there's an increase in myosin activity, in phosphorylated myosin activity, that is. Sorry about that, just had to figure that out real quick. And then, so this is their final figure, and they kind of showed a lot here. So I'm going to try to go through it the best I can, but it's a little confusing. So what they did was they used spheroids for this assay, which is like a little growth of cells. You can think of it, I mean, it's kind of like a little tumor, but they grew it in either physiological pH, which is, they use 7.4 throughout the paper, and then they induced acidosis, which they had as 6.6 throughout the paper. And what you see is that in all three of these, they look pretty similar in terms of like the H&E stain. But then when you have Piezo1 activated in the low pH condition, you get this fractionation of the spheroid. And so I'm going to, in E and F, they show that the Piezo1 active low pH condition results in significantly lower MTT reduction, which is a proliferation assay. So these cells aren't proliferating nearly as much as the control cells. And there's also an increase in an X and V positive cells. And X and V is a marker for apoptosis. So this fractionation is also representative of cell death. And then you also over here can see the manganese quench rates again for the different spheroids, and you see the rate is much lower in the intracellularly acidic conditions than in the neutral ones. So putting that all together, Dom and I made separate models and we tossed them in here. I kind of went with a more extrapolated version. I put it into a tumor, kind of like what they were showing from the spheroid assay, but I was showing that in, so if you have normal pH in the tumor exterior and you have acidosis in the tumor core, which is pretty representative of what they were showing in the paper, under control conditions not treated, the cell will, like the tumor core will stay together, and those cells won't undergo the apoptosis that we just saw in that last figure. But when you add the Yoda1 and increase Piezo1 activity, the cell, like the tumor core, would fractionate. And so I kind of took that as their like clinical takeaway of, obviously more studies, but it might be some way to fractionate like a tumor core.
Yeah, in a similar fashion, I kind of took the same route. I just showed a little bit more detail with the Piezo1 and the calcium. But obviously, there's a little bit more that we could have added with like the phosphorylation of myosin parts and other intracellular like actin cytoskeleton stuff. But there's kind of like a lot of little stuff thrown in there like that. So we kind of just focused on more of the physiological stuff. And you can see like in this pH 6.6 on the right, that I tried to show the pronation with like a really big H. It looks kind of weird, but I think it gets the point across that. And you can see that like calcium influx is lost versus on the left, you can see when Yoda1 is added, I probably could have labeled that little orange dot a little better for Yoda1. You can see that like calcium influx is then restored, but then you also get the fragmentation of the spheroid.
It's a great figure, Dom. Quit down talking
it.
So are there any questions? Because the next slide, I think, is our discussion questions. So if there's anything that wasn't clear at all, let us know. We can go back, or we can move on to the discussion. All right. Everyone good?
I'm good.
Okay. So we just had a few questions. Since we did learn about Piezo1 before, we kind of wanted to relate this paper back to the first one and bring the class full circle a little bit. So, yeah.
Yes, we're asking like what are some advantages or disadvantages of like the manganese quench method to detect Piezo1 activity versus like what we learned the first day class, which is like this, what's it called again?
The patch clamp.
Clamp method. Yeah, like obviously they're different and they're used in different situations, but I guess we can just like discuss like how exactly they may be used. So if we can show again, can you go back and show like how this one works?
Back to where?
Way back. So like you can see this one, like the manganese is outside the cell, and then we can stimulate it, and then we lose the fluorescence, and then yeah.
Mm-hmm. I'll go back. And then I had the question, what would be a good next experiment? What could this data inform, and what clinical impact might these data have?
Let's start with that first one.
Okay.
Let's just start there and then get the discussion going. So let's think about that one. And just if you have ideas, whenever, you can just start sharing. So one thing that I was thinking about, just this isn't necessarily the comparison, but it's an interesting thing to kind of learn a roundabout way about like the elements, you know, like I basically learned that manganese and calcium are similar elements, right? That's what this tells me. And so then what are the advantages and disadvantages, do we think? Like what's an advantage of like just looking at GFP signal, for example? You know, think about that.
Well, for GFP signal, I feel like it's just sort of easier to measure, like in terms of the equipment you need and just sort of how complicated it is to measure that. So.
Yeah, that's what I agree with. I think it'd just be way easier to visually see like what's happening and like whether it's being activated, and also it might be just less precise because I'm assuming the patch clamp method is very precise. Like you're literally measuring how like the change in flux with like one specific channel, so it can be much more precise. But yeah.
Well, not the biggest consideration. It's all GFP is also just something you can look at and have some intuitive understanding of what you're looking at, so it's a bit easier to explain.
Yeah, for sure.
Yeah, I was gonna say that too. Yeah.
Yeah, it's more accessible too. Like way more labs will have access to the technology of like the magnesium or the manganese quench, because they don't need like the whole patch clamp. Like they don't have to be an electrophys lab. So. Yeah. So is it chill if we move on to the next one?
Yep, definitely.
Cool. So I always like thinking about what would be a good next experiment, and what could this data inform and what clinical impact it might have. Especially the first part though, just like now that they have all of this data, what would be best to know next? Like if they could include one more thing on this paper, what might it be? Just food for thought again.
I have some just like fun. I think this paper is really fascinating to me. I'm really into like tumor migration and environment and stuff like that. This is really so I'm fascinated by this different microenvironments within the tumor and different pHs and how that's changing the stiffness and dispersal and all that. I guess I don't really have a question. I'm really into this idea. And I think it's interesting that we get this dispersal of the core, these core cells and these spheroids. And then I wonder if that, just by changing that pH and activating Piezo2 or 1, that can start to disperse our cells. That's great. Right. It's mind blowing to me. So that's all. I think it's a cool idea. I've not thought about the different pH environment of a tumor.
Anyone else? All right, well, we can also look at the last one. I just thought it would be worth mentioning Piezo2. If treated with an activator of Piezo2 as opposed to using Yoda 1 to activate Piezo1, would you expect to see similar results? Why or why not? This might be kind of out there, but a lot of like the activation of Piezo2 versus Piezo1 was in the first paper. So looking back at that one, I just thought it might be cool to, look at Piezo2 instead of Piezo1 on this paper as well.
I seem to remember that wasn't Piezo2 expressed in different cells, or am I misremembering that?
Yeah, I think it was expressed in some different cells, but it was also expressed differentially in the same cells. So it had a distinct signal. I think they were using the same cells. It would be
okay. I was just thinking that it might simply do nothing if it turns out that Piezo2 is barely expressed or not expressed at all in these specific cells, but I don't recall whether that's the case from that first paper.
Yeah, so my understanding was that it's still expressed, but it's expressed at a lower rate. So it might have like a slower rate of influx of calcium, might have a slower rate, or it might like let different things in because of the construction of it. So it is interesting to see. It'd be a cool experiment, I think. Anything else, Dom?
Nope. I think.
Cool.
Yeah, I think we're all good.
All right.
Thanks, guys.
Thank you. Let's get our mics unmuted and let's give them a round of applause. I'm the only one clapping.
We appreciate it.
Hey, I'm the only one who claps.